Without further ado, I'm going to pass the dice to Dr. Jordan. Please give him a warm welcome.
Lava, I was just getting ready to thank you and congratulate you on that great introduction, but that was a little bit too much disclosure there at the end, so we'll talk about that later. No, but thank you very much, and thank all of you for coming. I realize that it is very early in the morning, so I appreciate you being here. As you just heard from Lava, I'm going to tell you about some work, some collaborative work that we've been doing in Colombia for several years. So this is something that I'm really excited about. It involves collaborations with a number of different institutions. So before I tell you about the work, before we jump into the data, I just want to kind of try to motivate it and tell you a little bit about my journey. How is it that I ended up down in Colombia? So this work is very much aligned philosophically with a lot of the writing of Thomas Friedman. So I'm sure many of you are familiar with Friedman. He's a columnist for the New York Times. He's sort of a known commodity around here at Georgia Tech. He's visited here several times. And in his book, The World is Flat, he actually has a chapter on Georgia Tech. So he talks a lot about globalization. So Friedman talks about the challenges, but also the opportunities that come with globalization and how it is that we can take advantage of those. And so this statement from a very recent column of his really sort of underscores the opportunities that present themselves with these types of collaborations. So Friedman says that the biggest trend in the world today is the merger of globalization and the revolution in information technology. So this presents great opportunities for international collaborations in bioinformatics, which is my field, but also genomics, which is more and more a field that is driven by developments in information technology. So in that spirit, Georgia Tech a few years ago put out their strategic plan. So this is a big plan which tries to lay out the next 20, 25 years vision for the university. And there's 5 overarching goals in that plan, and no fewer than 3 of those 5 goals talk about international relations. And the 4th goal is explicitly about international relations. So the idea is that we really want to expand the global footprint, the influence of Georgia Tech around the world. And there's sort of 3 ways that we think about that. One is to sort of bring the world to Georgia Tech, if you will, expand the footprint of the world at Georgia Tech, and then project ourselves out into the world, so extend and leverage Georgia Tech's impact around the world. And finally, with respect to you students, we want to train and develop students that are really globally engaged and are ready and poised to take advantage of all these great opportunities that are arising. So here's a bunch of flags that sort of lay out how this has happened. And historically, Georgia Tech has gone, for reasons that make sense, to the west, to Europe, and to the east, to Asia. And what I'm trying to do is sort of go to our neighbors to the south with a particular emphasis on Colombia. So Colombia is really a key player in Latin America. It has a very dynamic and emerging economy, and there's a lot of great opportunities there. So a lot of these efforts, we've been doing this since around 2007, but things really picked up speed in 2012. So with support from the College of Sciences and also from Fulbright, I spent a tenure here in Cali, or a sabbatical rather, here in Cali, Colombia. So this is Colombia here, and Cali is in the southwest part of the country, close to the Pacific coast. So I was a visiting professor at 2 universities in Cali, Colombia, Universidad Libre, Universidad del Valle, and this is just some shots of Cali here. Here's Lava here after we summited a mountain outside of the town. And we've also gotten some other professors involved in this work. This is Joel Kotzka, who we're doing work on biofuels with sugarcane, and Fred Vanberg, who's been working a lot on the results I'll tell you about today in human genomics. So this was really a wonderful experience for me. A little bit before that, again, with support from the College of Sciences, right when the strategic plan came out, Paul Houston, who was dean at the time, and Terry Snell, who was chair of the School of Biology, said, well, we know you're going to Latin America a lot. Now we have the strategic plan, so why don't you try to formalize and deepen your efforts there and deepen your connections? So as a result of that, we launched this PanAmerican Bioinformatics Institute. So it's called an institute, but it's really more of a network. So it's a network of like-minded investigators in the US, and in Latin America, mostly in Colombia, but in different countries in Latin America. And we kind of have the goal, it's a little bit lofty and maybe a bit naive, but we basically want to take advantage or leverage biotechnology for public health and economic development across the Americas. And so we do that in three ways. We facilitate collaborative research, and that oftentimes is a matchmaking process whereby I look for labs in the United States that have technological capability that can be applied to problems that are specific to Latin America. We do student exchanges, so we bring a lot of students from Latin America here to labs at Georgia Tech and other institutions. That has gone very well. Something that I'm interested in doing to expand that would be to take students from here and send them down to labs in Latin America, and Lava is actually going to go do that this summer. And we do workshops and short courses in bioinformatics and genomics all over Latin America. So Lava also mentioned this Joint Genome Research Center. So this was a direct result of the sabbatical, my tenure as a visiting professor at Universidad del Valle. So we formed this last year. And again, the idea here is to use those sort of three arms, education, training, and research, to help facilitate the development of knowledge-based economy in Colombia and throughout Latin America. So that's sort of the background, and most recently we've been working with BIOS. So BIOS was just formed in late 2013. In October it opened. So BIOS is sort of Colombia's version of the NCBI. So I come from NCBI, that's the National Center for Biotechnology Information. It's sort of the world's premier institute for bioinformatics. And Colombia, and this is really the reason why I went down there originally, has decided to invest in the development of capacity in bioinformatics and genomics. So they brought me down there to do that. Part of a result of those efforts on the behalf of their government was the formation of BIOS. So BIOS is really driving to a great extent the work I'm going to talk about today. So we have this big collaborative project on human, clinical, and population genomics in Colombia. So the basic idea, I think that it's probably apparent, obvious to all of you, that genomic applications to health and medicine hold great promise. And when you apply genomic techniques to clinical issues, in order to do that, you typically need a big sort of background data set. So you need big surveys of the extent of genomic variation that exists among healthy individuals of any population in order to then be able to go and do a comparison to try to figure out what the genomic determinant of disease might be. And for obvious reasons, most of these big surveys and most of these clinical genomic studies are based on US and European populations. So what folks in Latin America realize is that we really need regional or country-specific surveys of the genomic variation that exists within and between those populations. And that's what we're trying to do, working with BIOS, working with Univalle in Colombia. In addition to doing those regional or country-specific surveys of genomic variation, it's also critical to develop local capacity in genome sequence and bioinformatics. And so to do that, again, we're involved in research, development in terms of developing software applications for bioinformatics and genomics, and training in human genomics and bioinformatics. So I'm very engaged in this, I'm passionate about it, but the ultimate goal is really to make ourselves dispensable. So if we do this capacity building correctly, then they will have and possess the local capacity to do everything that they want to do. Hopefully that'll take a while. Okay, so the first time I was invited to Colombia was in 2007. I knew very little about Latin America and almost nothing about Colombia. So you can imagine what I thought I was going to see, going on based on the reputation and what I was hearing in the news, and I was a little bit worried about going down there. But what I found was something very, very different from what I expected. And one of the things that was most fascinating to me and remains fascinating to me about Colombia was the extent of the human diversity that's there. So when you talk to biologists in Colombia, they often talk about biodiversity. So I think Colombia, after Costa Rica, is the second most biodiverse country on the planet. But in addition to the biodiversity, or one dimension of that biodiversity, is the human diversity. So Colombia has a very rich human diversity with three-way admixture, and I'll talk about exactly what I mean by admixture if you don't know that, but basically the people have a mix of ancestry, of African ancestry, Native American or Amerindian ancestry and European ancestry. And you can see that in the faces of these Colombians here. So here's just a breakdown of the demography. So the majority of the population are what anthropologists refer to as mestizos. That's a mix of Native American or Amerindian and European background. There's people with mainly European ancestry, African and European, and so on and so forth, and every permutation and combination thereof that you can think of. So another challenge to clinical applications to genomics, most of the surveys that are done rely on populations that are the most pure, for lack of a better word. So if you want to do clinical genomics, you typically start with a population of Europeans, and a really canonical population of Europeans, people that we know have all of their ancestry from Europe. If you're looking at Africans, you go to a very canonical African population where you're sure they're from Africa. But the fact is, in Colombia and really throughout the Americas, populations are very admixed. They have genomic ancestries from different continents. So that presents another challenge, but also an opportunity for these types of studies. Now, this is by no means the first study of this kind. So there's been a number of these studies. Most of them have not been at the genomic level. They're starting to get at the genomic level. And for the most part, what they're doing is just trying to take a snapshot of what the diversity is. So we want to do that too, and we're doing that. But what we want to do to extend that is then relate the standing genomic diversity in the populations, especially with respect to admixture, to determinants of health and disease, and how those are related to natural selection. So that's what we're going to try to do today. So this is an outline of what I'm going to talk about, and this is kind of the approach we used, and it's sort of a two-pronged approach. So the first thing, like those other studies I just showed, is to characterize the ancestry and admixture patterns in Colombian genomes. So we can do this at the continental level in terms of telling you what fraction of your genome comes from which part of the world. That's relatively straightforward. Something that's a little trickier, but we're making some headway on, is that we can also do it at the subcontinental level. So if you have African ancestry, we can say this part of your genome is likely from this part of Africa. We can do this genome-wide. We can do overall averages, but more interestingly, we can also find locus-specific patterns. So we can tell you for an individual gene in the genome, if you're Colombian, which part of the world that particular gene came from. In other words, what's the ancestry for that particular gene? And we can also do this for overall and sex-specific admixture. So it turns out that the male and female contributions to admixture patterns are quite different, and there's interesting historical reasons for that. So having done that, then what we want to do is relate the admixture patterns to health and natural selection. And this is really the new part here. And so we do this with a technique that we call admixture enrichment analysis. So we basically look amongst the population and try to characterize regions of the genome in the population that are enriched for ancestry from one or another continental population. And having done that, we then interrogate those anomalously admixed genomic regions to try to figure out if there's a relationship between that anomalous admixture pattern and, again, determinants of health, disease, and natural selection. So the basic idea is that we're trying to get at what I perceive to be a real paradox of human evolution. And what's that paradox? The idea is that humans evolved out of Africa about 60,000 to 100,000 years ago, okay? So for 99% of that human evolution, the rule was diversification and isolation. So populations migrated out of Africa, they diversified, they were isolated, and then there was assortative mating within populations. And this, of course, is what gave rise to all the different racial groups that we see today. So that was 99% of human evolution out of Africa. Within the last 500 years, which is really less than 1% of the evolution of the human species, this process was turned upside down. So now, instead of diversification, you have individuals from Europe coming over to the Americas, folks from Africa coming over to the Americas, mixing together, and now we have convergence and we have admixture. So what's happened here really in human evolution is that we have completely flipped the trajectory of evolution upside down, And we are, in a sense, creating genomes that are totally new. So the question, one of the big questions we're driven by is, what does it mean when you have these genomes that look completely different from human genomes that have been evolving for 99% of the trajectory of human evolution out of Africa? And that's what we're trying to get at. So to get at this, we need to sample two classes of genomes. We need to sample, so we're looking at a modern admixed population. So we need to sample what we consider to be the ancestral populations that gave rise to the modern population and the modern admixed populations. So again, the ancestral populations in the case of Colombia would be Europeans, Africans, and Native Americans. Now, it turns out that many of you probably know that Native Americans actually evolved from Asians relatively recently in human history. So they came across the Bering Strait here, they migrated south, and they populated all of the Americas, and that happened probably about 10,000 years ago. So it turns out that when you're looking at continental ancestry, Asian genomes are actually a very good surrogate for Latin American genomes, and we have a lot of Asian genomes, so we often use those to infer Amerindian ancestry. So these are just all of the data that we've sampled here to do this. So we have the ancestral populations from around the world from the three ancestral populations. We have admixed populations from the Americas. So these are all complete genome sequences or genotypes based on SNP arrays. We also do a lot of work with mitochondrial DNA and Y-DNA, again, to look at sex-specific admixture patterns. I'm not going to talk about that work today. And my background is a biologist. I do bioinformatics, but for me, a computer is just a tool to do biology. The computer scientists might have a different idea, but for me, this is really big data, right? So when we talk about the number of genomes, and this is very much a back-of-the-envelope calculation calculated by the number of base pairs in the genome, we get about 2.8 to 10 to the 11th points of data, okay, which Lava has calculated to be roughly equal to the number of stars in the galaxy. So for someone trained as a biologist like myself, this is heavy computational lifting, but that makes it exciting. Okay, so this is an important slide. This is sort of the essence of how it is that we do these techniques. So all of this admixture mapping, characterizing the patterns of admixture between the ancestral populations, is based on these SNPs that we refer to as ancestry informative markers. So this is an actual SNP. So here we have 3 Amerindian genome sequences, so ancestral from Native America, 3 European genome sequences, and 3 African genome sequences. And this SNP is considered to be an ancestry informative marker because it segregates between the ancestral African population and the European and Asian populations. So if you were an admixed individual and you had this SNP at this position, and then we would go out and look at the other SNPs in the neighborhood, then we could infer with some degree of confidence that that region of your genome is derived from the ancestral African population. We can extend that out across the whole genome and get those overall averages. So that's the way it works. So now let's look at this first at the continental level. So this is, if you take a number of genomes, so on the poles here we have both the Asian genomes and Native American genomes. So this is a Native American population from Bolivia, which has very little admixture, so it's a very sort of coherent population. This is another Native American population from Mexico, which has more European admixture. This is the European ancestral population, and this is African. So we do all against all genome sequence comparisons, and we get the pairwise distances between the genomes, and then we do a PCA analysis and project the relationships. Now, the Colombian genomes are shown here and here. So this is one population from Medellin. So Medellin is a city in the northwest part of the country, and this population looks kind of like what we consider the classical Colombian mestizo to look like, right? So they are quite admixed in the sense that they have a clear Native American dimension, also have an African dimension, but they cluster largely with the Europeans. More recently, we've been starting to work with Afro-Colombian genomes. So this is a complete genome sequence of an Afro-Colombian from Chocó, which is a state on the Pacific coast, which is about 80% Afro-Colombian. So this is starting to give us an interesting picture of the admixture, but it's very much incomplete. So I think you can kind of infer from the first picture of the Colombians that I showed you, along with the data from the demography, that what we really want to do is begin to fill in these axes along this dimension and along this dimension. So we have more work to do to fully characterize the diversity, but nevertheless, this is an interesting and unique pattern. And so going forward, we're going to focus on these genomes to try to understand, again, the relationship between admixture, selection, and health. So this is just having related them like that, then we can start to compute the overall averages for the populations. So this is the percent ancestry for any given genome. So it's difficult to see, but there are many, many genomes going down like this. This is the ancestral European population, ancestral Amerindian population, ancestral African population, and these are all of the Colombian individuals. And as you can see, they're very admixed, and this is just to give you an idea. The error level is up here. There's a little tiny bit of error, but this is obviously much more than the error. So if we zoom in on that, in green are shown all of those genomes from the Medellin population, along with the single Afro-Colombian. So this guy obviously looks quite different. So he has about 80% African ancestry, whereas most of the folks from Medellin have more European ancestry and more of a three-way admixture. So they have about, on average, 9 or 10% African ancestry and about 40% Native American and the rest European. Okay, so that's at the continental level. And now let's zoom down to the subcontinental level. And this is something that we're really looking at with respect to Afro-Colombian genomes. So we're getting more and more into this. And the question of, where do I come from is obviously an interesting question to everyone, but this is a particularly interesting question for Afro-descendants across the Americas. And the reason why is because there are not the kind of historical records available to them that there are for descendants, people with European ancestry. So African Americans here in the United States and throughout the Americas are really interested in applying these genomic techniques to figure out what their ancestry is. So we're working with our collaborators in Colombia, again, a lot on the Pacific coast where there's a big Afro-Colombian population, to try to figure out what is the subcontinental ancestry of the Afro-Colombian portion of their genomes. And so to do this, as I showed you earlier, we sample from a number of different tribes in Africa. So these are ancestral populations that come from the regions of Africa that we know from historical records were the sources of slaves that were brought to the New World, in particular brought to Colombia. And when we do that, so here's a similar PCA analysis, here's a tree, again, and here's our Afro-Colombian genome here. So we're working with a single point of data, we need more information, but it's quite interesting. So this guy does have admixture in the African part of his genome, but he really groups squarely with the Yoruba tribe in Nigeria. And this is consistent with the historical records, again, from where Afro-Colombians come from. It's also interesting because many African Americans have Yoruba ancestors. And in fact, the Yoruba tribe is the single biggest source of Africans in Africa and throughout the world. So probably about 20% of all Africans or African Americans are descended from the Yoruba tribe, and that holds for Colombia as well. We can also look at sex-specific ancestry with admixture, and this is very interesting. So we can do this with mitochondrial DNA, which goes through the female lineage, and the Y chromosome, which goes down the male lineage, but you can also do it by looking at autosomes. So what we're doing here is we're looking at the normalized ratio of the admixture that is found in the X chromosome versus the autosomes. And the reason why we do that is because the X chromosome spends more time in the female lineages, because females have 2 X's, males only have 1 X chromosome. So if you look at that ratio, you can see if there's a difference in the admixture pattern between the X chromosome, which would be consistent with female-specific ancestry, versus the autosomes, which would be consistent for male or paternal ancestry. And all Latin American countries have a very stark pattern, but Colombia has more than anything, where more than any of the other countries, where the maternal ancestry is very much biased towards the Amerindian or the Native American side, and the paternal ancestry is very much biased towards the European side. And this is actually quite different from what you would see if you would look at Americans in the US. And so the historical reason for this is that when Spaniards came to the New World, typically and initially it was men only. It was conquistadors that were coming over alone in search of gold and riches, and those conquistadors had families with Native American women, and you can see that here. Now in the Americas, groups of families came over together fleeing religious persecution, and consequently there was less of a bias-sex ratio, and you see a very different pattern. So there's interesting connections between what we know about the history and what we can see in the genomes of these individuals. Then finally we can get down, as I mentioned before, to the locus-specific level. That is, we can do these admixture analyses and we can begin to look at individual loci, individual genes, and make assessments about where these individual genes come from. So again, this goes back to the slide that I showed you before where we're looking at ancestry informative markers. But now what we're going to do, instead of just looking across the entire genome, is we're going to do what we call chromosome painting. Okay? So the idea is that we start with a number of ancestral genomes, or genomes that represent the ancestral African population. These are Yoruba genomes, so we have 87 of those. We have genomes that represent the ancestral Native American population, with the caveat that these genomes are actually from China. They're Asian, but they work very well, and you get the same results if you use Native American genomes. And then we have a European population. So if you recall that first ancestry informative marker, that SNP that segregates between the African and the European populations, if we had that SNP, and it's not only one SNP, it's the SNPs that occupy that environment, but for simplicity, that region of the genome. For simplicity, I'm only showing one SNP. Then we would paint that region of the genome blue. In other words, we would assign the African ancestry to that region of the genome, and then we just walk across the genome going along. And when you do that, these ancestry informative markers and their neighbors will tell you from which of the ancestral populations each segment of the genome is derived. So that's how that works, and this is how it looks. And so I'm just showing you a snapshot of a few of the genomes from the Medellin population. And these are some of the genomes that occupy the extreme. So in orange we have European ancestry, red Native American or Amerindian, and blue African. And so this is the individual in Medellin that has the most European ancestry. But even though this individual has the most European ancestry, you can see that there's quite a bit of admixture all across the genome. Okay? So here's the individual that has the highest level of Amerindian ancestry, and here's the individual that has the highest level of African ancestry. Now, as an aside, this is very interesting because people self-identify racially quite different in Latin America than they do in the US. So this population walking around Medellin consider themselves largely to be white. So if you were to ask this individual, what is your race? That person would most likely say, I'm white or I'm mestizo, despite the fact that they have 33% African ancestry. So all of these genomes are de-identified for very good reason. But one of the things we're exploring, and I'm not sure if we can do this, is to get volunteers who are willing to share their identity, like the Human Genome Project that's run out of Harvard, and just compare their phenotypes, how they look compared to these percentages. So I would be intrigued to have pictures of these individuals here, and I think that it wouldn't track like we think it would. But anyway, this is just the example of that type of chromosome painting. So at a fairly fine level of resolution, we can assign with confidence the ancestral regions across the genome. And then finally, that allows us to begin to tie in this process of admixture, this notion of creating genomes that are totally different from human genomes that have been evolving for 99% of human evolution, tying those patterns to determinants of health, disease, and natural selection. So again, the basic idea is that the last 1%, 500 years or so of human evolution has been characterized by this flipping upside down the paradigm, where now we have convergence and we have admixture. So what does that mean? What do these genomes look like? Okay, so what does it mean when genomes that have been separated for greater than 60,000 years are suddenly brought back together in the blink of an eye evolutionarily? And is that process of admixture somehow related to health and selection? So our working hypothesis is that there will be specific alleles, SNPs, that have evolved separately in these different human populations based on their regional specific utility. Okay, so we know that to be the case, but selection is a relatively slow process. So over many thousands of years in the ancestral homelands of these genomes, Africa, Asia, and the Americas, and Europe, alleles that provided selective utility, especially with respect to health and disease. So probably, or not probably, but definitely historically, the biggest single selection pressure on human genomes has been infectious disease. So humans have evolved a number of alleles that provide resistance to infectious disease, and this happens in a very regional specific manner. So many of you are probably familiar with the sickle cell trait that is prevalent among individuals of African descent, and it turns out that that allele is an allele that provides resistance to malaria. That's a classical case of regional specific selection for alleles that provide resistance to infectious disease. So we know all that to be the case, but then the idea is that you have these pre-evolved ancestral population specific alleles that when you create this new admixed population, that population, this is speaking a little bit anthropomorphically, but the population can select from among or choose from among those pre-selected alleles to construct genomes that would be the most fit. And again, they'd be selecting them based on their utility that has evolved over many thousands of years in the ancestral homelands. And we might expect that most of that selection pressure, again, would be based on resistance to infectious disease. And that would make sense in a tropical environment like Colombia or like much of the New World. So that's the basic idea. And the way that we try to get at that is with this population admixture enrichment approach. So we talked about all of those Colombian genomes that we have. We do the admixture mapping, so we paint their chromosomes with respect to the ancestral regions, and then we just stack them up on top of each other. And so the idea is that if, for example, in the Medellin population, you have on average 10% African ancestry, as you walk along the population of chromosomes, so we have 60 individuals, 120 chromosomes, you expect about, on average, 10% ancestry across all of those chromosomes, 10% African ancestry at any given region. And what we look is for segments that are enriched for African ancestry and those that are statistically significantly enriched. And we can also do that for Native American ancestry, and we can also do that for European ancestry. And then having done that, we begin to try to functionally, I'm using functionally sort of loosely because it relies on computational analysis of previous experimental work, but we functionally interrogate those genes to try to figure out, is there a connection between these regions that are enriched for ancestry from the different continental populations and selection, health, and disease. So if you do this for all of the genomes, this is a heat map which kind of gives you a sense of where the sweet spot is, where the averages converge. So we have, remember, 3 ancestral populations, Amerindian, African, and European. Here we only have 2 axes because the third one is dependent on those 2. And the average chromosomal segment, now not individual, it would fall right around here. So again, just below 10% African, right around 40% Native American, and right around 50% European. So what we're doing with respect to that ancestry enrichment analysis is looking for guys that fall out here. They have much more African ancestry than you expect by chance, ones that fall out here, much more Native American, and ones that fall down here, which would have more European ancestry than you expect to see by chance alone. So this is a Manhattan plot. I don't know if you all are familiar with these, but these are plots that are typically used in genome-wide association studies where you're looking for associations between individual SNPs and some disease. And what the Manhattan plot shows simply is the distribution of probability values, p-values, that any given SNP is associated with that disease. So we're using the same convention here, but we're using it differently. So the probability is derived rather from a trinomial. So this is just the probability that the ancestry enrichment that we observe at this position could be due to chance alone based on the background ancestry. And so what that allows us to do is find these individual peaks which are regions that are highly enriched for ancestral specific segments from Africa, from Europe, and from the Americas. And then we start to look at the functions. And so what happens, which is quite remarkable, or for us is that if you look at the enriched regions, so this is just a sort of classical GO or gene set enrichment, where you take those genes which occupy the top of that list of the most statistically significant ancestry enriched, and you just ask, are there functional categories that are overrepresented? Well, not unexpectedly, there are differences between the ancestry enriched segments from the different populations. This is hard to make out, but we'll get into more detail later. But what shows up over and over again are immune-related functions. So there's a lot of immune-related functions here in the African enriched regions, Native American, and European, but they are different sets of genes. So it looks like it's possible that two things might be going on. One is that the admixed genomes are sorting or somehow retaining alleles that are beneficial with respect, again, to mainly resistance to infectious disease, but doing so from different populations. That's one possibility, and these two possibilities are not mutually exclusive. The other one is that these admixed genomes are actually retaining alleles from different ancestral populations at the same loci to increase the diversity there, and a more diverse repertoire of immune-related genes might be expected to help you fight off disease more effectively. You could confront a more diverse repertoire of antigens in that way. And so an interesting example of this is the HLA locus here. So what we're looking at here are tracks from the UCSC genome browser. So I don't know if you are familiar looking at these type of things. So we're just reading across the genome here. And this is Lava's work. So he's made these custom tracks. So what he's done is just taken all of those painted chromosomes and just counted how many at any given region, stacking them up on the top of each other. How many are from Europe, again shown in yellow, how many are Asian, and how many are from the Americas. And then doing the enrichment analysis, he just gives you the log fold enrichment below. So this is sort of a zoomed out view of the HLA locus, which is part of the major histocompatibility locus, which is a very important locus, a very important region on chromosome 6 with respect to immune function. And there's a very clear enrichment of African ancestry there. This is zooming in a little more closely. And when you look in a little more closely, this falls at the gene HLA-B. Okay, so I mentioned this before. So this is the major histocompatibility locus. So it encodes receptors that present antigens to immune T cells. So the diversity of these receptors is directly related to their ability to bind and present a diverse repertoire of antigens, which is exactly what you would want to do if you wanted to be able to fight off a diverse set of infectious agents. Okay, so these are associated with many, many different diseases, and there's a tremendous diversity of HLA genes in African populations, and this is thought to be related to the pathogen richness in Africa. Again, the increased diversity provides an increased fitness advantage against diverse pathogens. So the idea is that maybe the ability to mix and match diverse African HLA alleles or alleles from other ancestral populations in the admixed population could also provide a selective advantage in the pathogen-rich environment of the New World. Okay? And this is just a plot showing pathogen richness. The darker you are, the more pathogen rich you are, and the allelic richness of HLA genes. Again, they correlate. And this is what's going on with our genomes. It turns out that yes, there is an enrichment of African ancestry at these HLA genes, but more than anything, what we see is an enrichment of combinations of alleles from different ancestral populations at the same locus. So this is an example with the HLA locus, okay? And so what we see are more cases like this than we would expect to see by chance alone. So again, the idea is that it might be that these admixed genomes have brought together assortments of alleles from different ancestral populations to encode a diverse repertoire of antigen-presenting receptors, which again would help them to fend off or fight against a diverse repertoire of infectious agents. And then finally, one more example, and this is sort of a counter example. So I told you about the ancestry enrichment analysis. This is a case of ancestry depletion. So here we're looking at the EDAR locus. So this is a zoomed out view, again, looking at the relative contributions of the different ancestral populations. Here's a zoomed in view of this locus, and here are the probabilities down here. So what you see at the EDAR locus is an underrepresentation, a statistically significant underrepresentation of Amerindian or Asian ancestry. Why is this interesting? So I don't know how many of you follow the literature on human selection, but there was a Cell paper published last year. Our own Fred Vanberg was one of the co-authors, and they talked about this EDAR locus. So EDAR is also a cell surface receptor, and it influences the development of hair follicles and subcutaneous glands. And there's very good evidence from that paper published last year by Vanberg and colleagues and previously that shows that there's been positive selection. So this says recent, but again, this is much older than the last 500 years. So the last few thousand years, recent positive selection at this locus in both Asian and Native American populations. And this selection was attributed to increased glandular secretions, which increases lubrication and reduces evaporation from the skin. Okay, so the idea is that this was a helpful trait that allowed these individuals to survive in cold and dry environments like Asia. So if we think about the New World and the tropics, the environment is exactly the opposite. So an adaptation to a cold and dry environment might be disadvantageous in the hotter and more humid tropical environment in the New World. And here's just a map of the temperatures around the world that underscores this. Again, this allele probably evolved here in East Asia and then was brought along with Native Americans as they populated the New World. And that might have been somewhat disadvantageous in the hot, humid environment of the New World, but when you had the opportunity to select from admixed alleles from different ancestral environments, those disadvantageous alleles were displaced by alleles that allowed for more evaporation, more sweat from both the European and the African ancestral populations. And then there's just a number of different alleles, again, many of them related to immune function that we're quite interested in looking at, that are consistent with this idea that there's been an anomalous pattern of enrichment for different ancestral populations in such a way as possibly to facilitate the ability of populations in the New Worlds to fight off infectious disease. So that's what we're looking at. One of the things we could use some help with, we are bioinformaticists, is sort of digging into these gene lists and trying to make functional sense out of them. So that's where we are right now. We have a lot more to do, but it's been a very rewarding and interesting collaboration. So just to recap, Colombians have highly admixed genomes, three-way admixture with African, Amerindian, and European ancestry. This ancestry can be assigned both at the continental and subcontinental levels. We're looking in much more deeply on this problem right now, trying to combine genetic ancestry with historical records using a Bayesian framework. That's something we're very excited about that I didn't have time to talk about today. The admixture patterns are very sex-specific. In Colombia, many Latin Americas, but more so in Colombia than anywhere else, the maternal lineage is dominated by contributions from Amerindian side, whereas the paternal lineage is more European. When you look across the genome, there's substantial and highly statistically significant enrichment of ancestry-specific loci, okay? And it's possible that alleles that reside in those enriched regions were pre-selected in ancestral regions, and they have been differentially retained for their utility in the New World. And this is, a lot of this has to do with immune function. So having said all that, I want to thank my collaborators in Colombia and here in the States. Again, this was really all made possible by Fulbright, which provided me support for my sabbatical leave in Colombia. We're all working together under the umbrella of the PanAmerican Bioinformatics Network. In Cali, I worked with Unilibre and Univalle, and we're really excited about our new work with BIOS. This is Augusto Valderrama, he's from Unilibre. This is Mauricio Rodriguez, he is the director of BIOS. Fred Vanberg, here at Georgia Tech was involved in all of this work. Andy Conley was a former grad student of mine that really sort of pioneered this work in the lab and has gone on to a research scientist position at Cedars-Sinai in LA. Charles Wigington is a current PhD student in Joshua Weitz's lab who started a lot of this work as a rotation project. And of course, there's the indomitable Lava, who I get emails from at all hours of the night, and he is responsible for these great slides. I could never make slides that look that beautiful, and he's really, really driving this work forward, and he's very excited about going down to Colombia this summer to keep that going. So thank you very much for your attention. I'd be happy to take questions. Yes?
I'm sorry if this is a silly question. It combines two of my least favorite things, statistics and immunology, but are people really best served by a diversity of their HLA repertoire? Or, I mean, because I would imagine that in certain physical locations, the pathogens may be unique to a physical location, and given that you have a limited set of HLA genes that you could encode, if you have diversity, I might think that you have a bunch of genes which have limited utility against the local pathogens that might present themselves.
Yeah, it's a very interesting question. It's a different way of looking at it. I think that the way that the field thinks about it, at least for this moment, and going back to that map, we can actually go back and look at that, is that the general correlation that's been seen around the world is that pathogen richness is very much correlated with the diversity that you see. So, but your point is well taken, because there are certainly regional specific pathogens that you think you might want to keep that particular HLA allele to confront those. So maybe both are going on, but the general consensus at this moment is that yes, diverse is better with respect to these type of antigen presenting receptors.
Do we know anything about, I'm sorry.
No, it's fine.
Clarifying question related to that.
Yeah.
Is that because the number of pathogens always exceeds the number of defenses within code? If there are 1 million pathogens and 1,000 possible defenses, then more diversity will always help. Whereas if there are 1 million pathogens and 2 million defenses, then diversity doesn't help.
Yeah, again, that's a very interesting point, and full disclosure, I don't know what the ratio of the number of pathogens is to the number of alleles. But what you're saying certainly makes sense. But again, the general rule that's been seen is that the more diverse, the better you are able to confront a diverse repertoire of pathogens. Yeah. John?
So to follow this up, pathogens are vectored by people. So if you come into an admixture, they're bringing with them variable pathogens. So it may be just an equilibrium kind of thing is maintained between the number of pathogens. That is, it's not an independent, it's not like you're coming into an environment with more pathogens. The fact that you're at admixture, you're bringing more pathogens into a single place.
Right, so that's very interesting, and I never considered that. So the selection pressure in that case would be not so much related to the tropical environment of the New World, but rather the fact that you're bringing all those people together. And we know, in fact, in history that when Europeans brought over their diseases to the New World, that Native Americans were not able to fight off many of those diseases. So that's entirely consistent with that. And that's a really interesting, different way of thinking about it that we had not considered. Yeah.
I was going to ask about that very idea. So the fact that you're using Asian populations as representative of Amerindian populations would miss a recent event like this, right? There were, large swaths of the Amerindian population were wiped out by European-oriented diseases. So have you detected that event?
Well, in a certain sense, we are seeing that in the admixed populations. So, for example, when we talk about sex-specific ancestry and admixture, if you go somewhere like Puerto Rico, there are no more Tainos walking around, right? But if you look in the genomes of Puerto Ricans, there are the traces of the Tainos. With respect to sex-specific ancestry, it's almost entirely on the maternal side. So in a sense, the ancestral populations might not show that, but the admixed populations do. But just to go back a step, of course, we would love to have a big collection of Native American genomes, but it's very hard to find Native American populations that are not admixed. Yeah. Good. Do you have? Yeah.
Yeah, this is a bigger picture question. First of all, I really enjoyed your talk.
Thank you.
But it's always dangerous to predict the future. But could you just comment on where we're headed in terms of using genomic data to predict disease and impact healthcare?
Yeah.
You know, the controversy over FDA regulations.
Right. So this is definitely a big picture question, where we're going. Many of you may not know that, so 23andMe, the company that's related to Google that was providing these genetic tests, and they were really big on Ancestry and they did a great job, but another thing they did is they provided you with all of your risk alleles. And those risk alleles were Ancestry dependent. And they told you, well, you have a risk of getting this disease or that disease. And the FDA shut them down, and I think that was January of this year. And so, you know, there's sort of two camps in the field. So one camp says, well, this information, and this is sort of the FDA perspective, is very delicate, and people that are misinformed, it's hard to understand that even for geneticists, how you calculate a risk factor for an allele, especially given the genetic combination. People are just not equipped to interpret that, and nor are physicians, so it's best not to put that information in their hand. I disagree with that. So I'm very much, you know, bioinformatics is very much an open source philosophy field. So I think you should put the information into the hands of the people. I think that computing those risk alleles will get better and better with more and more information. I think that patients should be informed, and they should go to their physicians and say, hey, what does this mean? And the physicians in turn should be pushed to be able to wrap their mind around that data and figure that out. And I suspect, well, I should be careful, but I wonder about the FDA's decision if there was some pressure from physician groups coming from that end wanting to control the information. That doesn't really answer your question as to where we're going, but if I were to make a guess, I would say the way we would be going would be to more and more openness with this type of information, with all the caveats that you have to be very careful with it. Yeah. Yes?
This map exhibits where you're blocking things by country, or not blocking, you're grouping things by country or continent or subcontinent. But trying to get all one color in terms of pathogen risk. But aren't the areas in like southeastern China more pathogen rich than, say, Mongolia?
Absolutely. So in that sense, this would be a rather coarse level view. And that gets back to the question that was asked earlier about very regional specific pathogens. And certainly they are there. Yeah.
Do you have any way to address this other than taking more data?
This is not our work, but I'm sure that people, or I suspect that people have tried to take more of a fine-grained look at that. So the answer to that would just be to do more regional specific surveys of pathogen richness and the specific identity of pathogens that occupy different regions. Yeah. Is that Emery? Oh. Hi, Jesse. Do you have a question?
Yes.
Okay.
So I was just wondering, going back to those climate-specific genes, what is it about 500 years of evolution, so maybe like 25 generations, would that be sufficient to select out genes that weren't kind of critical for survival against pathogens?
Right, so that's a very good question. So the time scale here is very, very short. So this is not, and this is a little tricky, this is not selection as we typically think about it, where these things have evolved over many thousands of years to arise from a mutation that is in very low frequency and then to be swept to high frequency based on the utility. So this is more of a picking and choosing from alleles that have been pre-selected in the ancestral regions over many thousands of years. And we think that that process can, and in fact has happened over a much faster time frame, because you're just sort of picking and choosing from those alleles, rather than evolving them de novo over many thousands of years.
But I guess then how would it go to choose the allele from a certain ancestry?
I didn't catch that. Can you repeat that?
Sorry, so how would it decide to choose the allele from a specific ancestry?
So the idea is that, and again, when we speak about this, I probably tend to anthropomorphize a little bit too much. So it's not a conscious selection, but imagine if you are in a pathogen-rich environment, be that, related to the tropics, or the fact that you're bringing together people with different pathogens that have never been together, that individuals that retained the loci from the ancestral regions, or the combinations of loci that allowed them to better fight off those pathogens, would have higher fitness, and their offspring would have higher fitness. So it is a classic differential reproductive success argument, but it's different in the time frame, in the sense that you're choosing from pre-selected alleles. Yeah. Good. All right. Thank you very much for your attention.